So after the initiation of the transcription, the elongation is also mediated by different transcription factors, okay? Especially the general transcription factors that help the starting of the transcription. They are dissociated because the RNA polymerase will leave that promoter elements. And then when that happens, a different set of transcription factors like factor 2S and SPTs, they are required for RNA processing. So when you say RNA processing, what does that mean? In the eukaryotic cells, as soon as transcription is happening, then the 5' end will be methylated with the G, guanine, and then at the end, the 3' end will be polyadenylated. Multiple A's, extra A's are added. And then the splicing will take place to remove introns, and those things are considered as RNA processing. So that will happen almost at the same time. Okay, so these transcription factors, SPT5 and factor 2S, are required for the processing. That's what it means. Okay, and the enzymes involved in RNA processing are recruited to the C terminus of the polymerase number 2. Okay, so like a capping enzyme as you're showing here, so 5' end capping, and then splicing machinery. Okay, again, they are recruited by the C terminus of the RNA polymerase, and this is polyadenylation factors also recruited by the C terminus. That's what is shown up there. Okay. So phosphorylation sites of C terminus tail area. So promoter escape, capping, splicing, and those are involved with the phosphorylation of the C terminus. That's what it means. The kinase A, especially the TEFB, can phosphorylate the serine at the position number 2. Okay, so it's this one here. Which correlates the elongation. Okay, and then TFB also phosphorylates elongation factor, SPT5. That's a NOS-G homolog protein, and the TATSF1s. Understand, so different set of transcription factors are involved in the elongation process. That's what it says. And these SPT5 and NOS-G factors can also bind between the factor 2B and the SPT5 area. They may have some regulatory function during the elongation. Okay, and then some RNA polymerase enzymes are already in the promoter sequence area, so they're just staying there as an inactive form. In that case, this kinase enzyme, TFB, when it phosphorylates the polymerase, they can activate the polymerase. So it can start the elongation. Okay, so different factors can do the similar thing. They can activate the polymerase so it can start the transcription continuously. That's elongation start. Factor 2S can also work as the elongation factor that can activate the polymerase enzyme. That's what it says. And factor 2S also contribute reading by polymerase by stimulating RNase activity of polymerase to remove misincorporated bases if there was any. Okay, and then FACT, which stands for facilitates chromatin transcription, are involved in reassembly of nucleosome structure ahead of RNA polymerase. So what it means is, the histone octamer structure. So the histone octamer wraps around DNA, the helical structure, and then they make nucleosome, right? So that's kind of compact DNA structure. So if the RNA polymerase will continuously work on its transcription process, those compact histone octamer structure needs to be opened up, dissociated, right? So that dissociation is mediated by this FACT, facilitates chromatin transcription kind of complex. Okay, and they come in as a heterodimer made up of two proteins, SPT16 and SSRP1. So those two proteins, okay, like this, okay, they come and help RNA polymerase to deal with the histone proteins in the nucleosome structure. So especially SPT16 binds to histone protein A and B dimers, and the other protein SSLP binds to histone protein 3 and 4. So tetramer, so that they kind of dismantle the histone nucleosome structure, octamer structure. So by doing so, your RNA polymerase can continue its transcription. So these proteins work ahead of the RNA polymerase. So DNA structure can be opened up a little bit. So that's what it means. Okay. And now let's talk about RNA processing, and that's happening almost simultaneously right after the initiation elongation of the transcription. Okay, so like I said before, processing includes the 5' end of the RNA being G-methylated, capped, and the 3' end will be polyadenylated. And of course, intron splicing is involved. So there are 3 major RNA processing steps involved. Okay, among those, so 5' end G capping with the methylated G. Okay, let's see how that happens. So SPT protein plays an important role. So that protein recruits 5' capping enzyme to the C-terminal domain of the RNA polymerase, especially the phosphorylation happens to the serine 5 of the RNA polymerase. Okay, and then when that happens, a phosphate is removed from 5' end of RNA. So as soon as it is emerged from the polymerase, so when RNA is coming out of the RNA, the polymerase through the transcription. So what it means is, so here you see there are 3 phosphate on the 5' end, right? So one of the 3 is removed. That's what it means. Okay, one phosphate is removed. Okay, and then G nucleotide, okay, triphosphate nucleotide is brought in by an enzyme, of course, capping enzyme, and then GMP, especially, pyrophosphate will be removed, and then it's going to become a GMP, and GMP is added to this end. So between these 2 phosphate and 1 phosphate of the G nucleotide, so you see that addition of the extra nucleotide, GMP addition right there. Okay, and then after that, the base of the guanine is methylated. Okay, methyl group is attached to this end. Okay, so that is done by the capping enzyme and addition of the G, and then once that's done, methylated G is added. Then yes, that enzyme is coming off from this complex. Okay, and then serine 5 of the RNA polymerase is dephosphorylated after that. So that's the methylated G capping of the 5' end. That's what it means. So you may wonder, why does that happen? The thing is, in the eukaryotic cells, transcription happens, and then after transcript is made, that RNA should be traveling from the site of transcription, which is the inside of nucleus, and to the place where ribosomes are located. Normally that's the endoplasmic reticulum. So there's a time gap between the transcription and the translation, because your transcript should travel. So there's a time gap. So during the time gap, a certain period of time, your RNA molecule, the transcript mRNA, should be protected. Otherwise, it can be degraded through a lot of enzymes. So this 5' end capping, G methylated capping, can protect your DNA from the degradation by RNA chewing enzyme. So it's a kind of protective mechanism, if you will. Okay. Serine 2 phosphorylation happening at the C-terminal domain of RNA polymerase can recruit RNA splicing enzymes. So RNA splicing means what? The introns that are stuck in the middle of the the coding sequence will be removed. So that is mediated by this process. Okay, and then another one is the polyadenylation happening on the C-terminus of your RNA. Okay, so the polyadenylation is mediated by these 2 proteins, CPSF, which stands for cleavage and phosphorylation specificity factor, and CSTF, cleavage stimulation factor. Okay, those 2 are carried by C-terminus domain of the polymerase. Of course, they will transfer to the poly-A signal once it is transcribed. So that is described here. So they used to be on here on the C-terminal domain of the polymerase, but they will be transferred to the probably 3' end area of your RNA. Okay, once they are transferred, and then they cleavage, they cut off the RNA at 3' end, and then add about 200 extra A's, adenine nucleotides to the 3' end. That's what it means by polyadenylation. Multiple extra A's are added. Okay, see the process. So a poly-A signal sequence is there in the RNA itself, and then RNA cleavage is happening. Okay, and then the CSTF that follows up. Okay, and then CPSF is recruiting poly-A polymerase enzyme. They can add extra A's, about 200 A's are added. Okay, so that's what is described here. Additional poly-A binding protein is involved in that as well. So it's a polyadenylation of your RNA transcript that's described here. So your textbook talks about two different transcriptional termination mechanisms in eukaryotic cells. One is depending on RNA's RET1 enzyme-dependent process. So what it shows is that enzyme can chew up the RNA that's transcribed after the completion of successful transcription. So if you look at this diagram, that RET1 and HXME complex is part of that RNA polymerase complex. But as soon as your transcription is done, so poly-A signal, that area is transcribed, the full transcript is the dissociated, right? But your RNA polymerase may not know that happened, so it may pass that area and start on a transcription of another part, another gene after that position. Then a new transcript may be coming out. Okay, at that point, that 5' end of the new transcript is not G-methylated, right? So there's no protection for that enzyme. So like I said, 5' end G-methylation is kind of a cap that protects your RNA integrity from RNA chewing enzyme, right? But this area is not methylated or G-capped. So that enzyme is that area is very prone to degradation. So at that time, RNA-RET1 comes along and get rid of that RNA transcripts that's coming off from RNA polymerase. So since it keeps chewing on, then that gives a kind of signal for RNA polymerase. Okay, I'm doing something wrong, and I should not make this RNA transcript that's just being degraded right away. So RNA polymerase recognize that as the end of the transcription, and it just folds up. That's RNA-RET1 mediated transcription termination. Okay, another one is another termination mechanism is allosteric termination. That is, depending on the polyadenylation of your full transcript. Okay, and then dissociation of that, and that process some kind of gives some kind of signal to RNA polymerase, which was transcribing. So dissociation and the polyadenylation of this full transcript kind of gives a signal for RNA polymerase. Okay, I did something that is meaningful. I finished my transcription, and then there's allosteric conformational change, meaning there's a signal transduction happens, so RNA polymerase changes structure, so it follows up that way. So it's a conformational change of the polymerase. Okay, that due to that, polymerase follows up, and that's the second mechanism that's explained in your textbook. Okay.